Hi, today I'll be reviewing the article Spindle Asymmetry Drives Non-Mendelian Chromosome Segregation. As you can see in the introduction, this is what I'm going to be talking about today, and here's a background story of the article. Selfish elements are often found to violate Mendel's law of segregation by the help of CDC4/2 signaling from the cortex, in order to increase the chances of inheritance into the next generation. This experiment is aimed to reveal how selfish element increases its chance of getting transmitted to an egg. These include spore killing, the CDC4/2 signaling from the cortical polarization directed by chromosome. Here I will be presenting the 5 experiments performed in this article. Number 1, the oocyte spindle in post-translational modification on microtubules. By placing the oocytes next to the cortex, this allows cytokinesis to produce a large egg and a small polar body. Screening mouse oocytes for marker as shown in figure 1b. This states that tyrosinated and detyrosinated alpha tubulin show complementary asymmetry while beta tubulin did not. Number 2, the effects of spindles when placed on the cortical and neocortex. In late metaphase 1, spindles were found to be asymmetric when placed close to the cortex. It was found that asymmetry depends on the time and proximity of the cortical. In order to test the spindle must be manipulated by treating the oocytes with cytochalysis or the CCB before the maturation stage to allow inhibition of actin polymerization. The results indicate cortical proximity, which induces asymmetry. This suggests that spindle creates higher level of tyrosine alpha tubulin, and therefore these results suggest that the cortex directly results microtubules to induce asymmetry within the spindle. Number 3, the polarized cortex. Tests were performed to find out whether the actin cap contributes to asymmetry spindle by inhibiting actin nucleating complex by small molecule inhibitor CK666, which stops the actin formation. Therefore, this suggests that the active CDC4/2 GTP phase is sufficient to increase alpha tubulin tyrosination and required for spindle asymmetry independent of actin cap formation. Number 4, asymmetry localization of active CDC4/2 mechanism to induce asymmetry. Autogenetic targets was developed to target the active CDC4/2 to one pole of the center. This result suggests that localizing CDC4/2 activity induces asymmetry within the spindle. Although some factors may cause weaker asymmetry, but by this approach compared to the asymmetry observed normally in the spindle near the cortex. Number 5, to measure the bias orientation of selfish centromere, the strongest centromere needs to be segregated by fluorescently tagged CENPB in a live cell. The strongest centromere must contain high levels of both inner and outer kinetochore proteins. Late metaphase shows how the strongest centromere is being favored by reorientation or flipping of stronger centromeres from the cortical straight to the egg, while the spindle is positioned asymmetrically. Further experiment was then performed by manipulation of the tyrosination levels by modulating the expression of tubulin tyrosine ligase, or the TTL. This catalyzes alpha tubulin overexpression of, in this case, destabilizing the spindle in the microtubules, as can be seen in figure 4c. This indicates that stronger centromeres interact with two different sides of the spindles but prefer to be on the egg pole. In a conclusion, as a result, it was found that asymmetry within the spindle is an essential factor in driving the meiotic drive and the first mechanism insight to how the signal from cell cortex regulate the microtubules to induce the spindle asymmetry and discover how the selfish centromeres are able to interact with the asymmetry spindles, as shown in figure 4e here. This is due to the cortical side of the spindle being found in the polar body. This explains the spindle asymmetry being consistently orientated to the cell fate, which provides spatial cues which guides the segregation of selfish elements. The cortical signals are a product of cortical polarization and is directed by chromosome that is positioned near the cortex. This positioning is incredibly important to the female meiosis as it allows the highly asymmetric division. and this allows the selfish drive element to exploit the asymmetry inherent in the female meiosis to increases the chances of them getting transmitted into the next generation.